This is a recording, a tour of two types of ganglion, sympathetic and sensory ganglion cells, a part of the nervous system. And so first I'll click on the sympathetic ganglion, and these ganglion lie along the spinal cord and have the cell bodies for the sympathetic nerves that control blood vessel diameter as well as control digestive systems. Actually, the sympathetic system kind of shuts down the digestive tract. So I have an annotation here that guides you to the cell bodies of these sympathetic neurons. And one of the features of a sympathetic neuron, of course, here's the cytoplasm, and it's fairly basophilic because it's loaded with rough endoplasmic reticulum, because all neurons are producing protein all the time. The transmitter substances as well as the enzymes that destroy the transmitter substance at the ends of the nerve fibers, at the terminations and synapses. But the nucleus is often located off to one side of the cell. That's one way. We're really not holding you responsible for that. We just want you to recognize when you see a neuron cell body, and also whether it's a motor neuron from the spinal cord or, say, in the brain, the pyramidal cells, and you'll see that in the lectures and the lab. Just looking around briefly, here's another specimen here, and if we enlarge this, you can almost barely see a nucleolus. If I go a step further in enlargement, you can see that's the nucleolus, this is the nucleus, and this is the cytoplasm that has patches of dark blue and light blue, not very well stained, but those are that's the endoplasmic reticulum, which is the factory for producing protein. And out here, all of these cells belong to the neuroglia, most of which would be Schwann cells in this particular situation, and the satellite cells around, which is never really that well defined in a sympathetic neuron or ganglion. So you can look around and see more examples. Here's another good example of a sympathetic neuron here. Okay, so I'm going to switch over to the other specimen now, which is a sensory ganglion, and these are located along the spinal cord, and they're part of each of the spinal nerves, and it's the sensory component, because each spinal nerve has a sensory and motor root, which join together to form a spinal nerve that then goes out and innervates the skeletal muscle of the body all over the body, but also receives and transmits touch and feel and heat and cold and things from all of our skin, etc. So actually this second annotation should be up on the first one, but it's actually just and I see there is a link to an HTTP here, so I've got to get rid of that. I didn't intend for that to be, but if I click out here, we can enlarge that. And so these are the locations of the cell bodies of the sensory neurons, and another bundle here. And then what we have in between are nerve fiber bundles that are connected to these cell bodies. If I click up here, then you will see the detail within that, and you see that most of the nuclei are in the center of the cell, and you can see, if I enlarge this a bit more, that you can see the patches of blue and lighter blue or pink, and that's the clumps of the endoplasmic reticulum. But you can also see the satellite cells around here, which really are modified Schwann cells that are coming up over, extending from the nerve fibers that are connected to these neurons. So study that and combine what you learned here with WebMic and the lecture. And then I'm going to click on the last annotation, which is highlighting 3 nerve fascicles. and a couple blood vessels. You'll soon learn in the next module that these would be venules, very thin-walled, and actually this is probably a monocyte in here with erythrocytes. But all these cells in here are Schwann cells, and the structures between, if I enlarge that a little bit, would be like these little thin strands here. That would be the axis cylinder, and so any gaps would be some myelinated, because some of these sensory neurons are myelinated, whereas the sympathetic are unmyelinated. Okay, so I think that concludes a highlight of these two specimens.